So we recently just discussed monosaccharides as well as disaccharides, and now we're going to discuss polysaccharides. And a polysaccharide is a very large carbohydrate molecule consisting of a few hundred to a few thousand monosaccharides joined by glycosidic linkages. And polysaccharides have two important roles, which are energy storage and structural support. So first we're going to discuss energy storage, and then we'll move on later to structural support. So the first kind of polysaccharide that functions for energy storage is glycogen. And so, inside each of our cells, we store glucose in the form of glycogen. And glycogen is a homopolymer. And what that means is glycogen is a polysaccharide in which all of the monosaccharides are the same. Now, in glycogen, we have two different types of bonds. We have the more common alpha 1,4 glycosidic bond and the less common alpha 1,6 glycosidic bond. And we call this bond right here, this bond, we call this bond an alpha 1,4 bond because it exists between the first carbon of this glucose, so this first carbon, and the fourth carbon on this glucose. And these glucoses have an alpha configuration, meaning that this bond right here, this bond, points downward compared to this bond which points upward. And so this alpha 1,4 linkage creates essentially a helical structure in glycogen. Now, the less common alpha 1,6 glycosidic bond, which occurs about every 10 glucose monomers, has a branching point along the helical structure of glucose. And this is a alpha 1,6 glycosidic bond because it occurs between the first carbon on this glucose and the sixth carbon on this glucose. And so again, because these are the alpha anomers of glucose, because this bond right here points downward compared to this bond which points upward, this bond is an alpha 1,6. And this one is a alpha 1,4. So now let's discuss starch. And so remember that glycogen functions for energy storage in animals and humans. Plants, however, they do not have glycogen. And so the molecule that functions for energy storage in plants is called starch. And starch is also the most common polysaccharide in plants. So starch comes in two forms, amylose and amylopectin. So let's first begin by discussing amylose. Now, amylose is an unbranched polysaccharide that consists of glucose monosaccharides. And these individual glucose monomers are connected by only one type of bond, which is the alpha 1,4 glycosidic bond. And remember that it's going to be the alpha 1,4 glycosidic bond because the bond exists between the first carbon of this glucose and the fourth carbon on this glucose. And we have the alpha because we have the alpha anomers of glucose, since this bond right here points downward and this bond right here points upward. However, the only difference between glycogen and amylose is that amylose doesn't have those alpha 1,6 glycosidic bonds because amylose is unbranched. And just like how we mentioned that glycogen has a helical structure because it contains alpha 1,4 glycosidic bonds, Amylose will also have the same helical structure because it has the same alpha 1,4 glycosidic bonds. Now let's move on to amylopectin. So this is amylose. Now amylopectin is essentially the same as glycogen, and this is because amylopectin contains both alpha 1,4 glycosidic bonds that we see here and the alpha 1,6 glycosidic bonds, which are the same bonds present in glycogen. Now the only difference between amylopectin and glycogen is that the alpha 1,6 glycosidic bonds in amylopectin occur much less frequently than those in glycogen. Now recall that these alpha 1,6 glycosidic bonds in glycogen occur 1 every 10 glucose monomers. However, in amylopectin, it occurs once every 30 glucose monosaccharides. So now let's move on to the second function of polysaccharides, which is to help with structural support. Now the first kind of polysaccharide that functions for structural support is cellulose. Now, one of the most common types of organic compounds on Earth is cellulose, and cellulose plays a role in structure in plants. Now, glucose monosaccharides are connected by beta 1,4 glycosidic bonds, and this means that cellulose does not have a helical structure, but instead, these beta 1,4 linkages allow cellulose to form very long, linear fibers that can stack upon each other via a particular type of non-covalent interaction called the hydrogen bond. Now, as a result of this linear structure, cellulose is ideal for providing structure, protection, and support to plants. Now, these bonds are called beta 1,4 glycosidic bonds because it occurs between this first carbon, and this fourth carbon right here. And this glucose that I've drawn is the beta anomer of glucose because the orientation of this first carbon, this first bond, is pointing upward. This bond is pointing upward, and this bond is also pointing upward. So we have a beta 1,4 glycosidic linkage or bond in cellulose. Now the second kind of molecule or polysaccharide that functions for structural support is chitin. And chitin is a structural polymer that is used by animals in the phylum Arthropoda to build their exoskeleton. And an exoskeleton is a hard case that surrounds an animal. And so some of these animals that have chitin in their exoskeleton include crabs, spiders, scorpions, centipedes, etc. Now chitin is structurally identical to beta glucose, except that the hydroxyl group on the second carbon is replaced by a nitrogen containing group called the N-acetyl group. So this is the nitrogen containing group, and it's called the N-acetyl group. And from organic chemistry, we know that an acetyl group consists of a methyl group single bonded to a carbonyl group. And so an N-acetyl group is an acetyl group attached to a nitrogen atom. And so we can see this is our methyl group right here, and then this is our carbonyl group in this lime color, and then we have the nitrogen in green. And so this is our N-acetyl group. And in these animals, in the phylum Arthropoda, their exoskeleton is made up of chitin, and this chitin is found in a layer of proteins. And originally, the exoskeleton is soft and flexible, but it becomes harder when these proteins are chemically linked to each other, as in insects, or when the exoskeleton is encrusted with calcium carbonate, as in crabs. Also, I want to add that chitin is not only found in the animals of the phylum Arthropoda, but it's also found in fungi to help them build their cell walls. And so these are the main functions of polysaccharides, glycogen, starch, cellulose, and chitin in both animals as well as plants.